Morena Tato, ko David Chanier Aho. So I'm David Chanier from Plant and Food Research, and today I'm hosting the Genomics Aotearoa seminar. It's actually a fantastic pleasure for me to host this seminar today, because as for once, I'm hosting a seminar from someone who's talking from my Whenua, my land of origin. So our guest speaker comes from Bordeaux in France, and that's where I did my studies and PhD in that lab. And in fact, I was visiting that lab last year for a sabbatical. So it's great to see a guest speaker talking today. So I'll give you an introduction to Santiago González-Martínez. So Santiago is a research leader in one of the leading lab, probably the biggest lab working on forestries, genomics, and a big focus of this lab called BIOGECO is about understanding the genomic control of adaptation of forestries to biotic and abiotic stress, and in particular climate change. So I've met Santi quite a few years ago, back in 2000. Actually, it dates back to our days at UC Davis, actually. So it ages us, but it's Santi is a world leader in this area. So it's a great honor to have you talking today, especially on a late evening in France. So without further introduction, Santi, the floor is yours. And just before you start, Santi, sorry, if you got any question, as usual, just put the questions in the chat, and we'll do the Q&A at the end of the presentation. Okay. Go ahead. Sorry.
Okay. Thank you so much, David. I'm very happy to be here tonight for me, presenting some of our recent work. So as David said, I work in Bordeaux at the INRAE in France, and I'm leading a research line on ecological genomics of forestries, mostly. I'm working also with other plants, but mostly with forestries. And I'm going to talk today about how to use genomics to assess adaptation or maladaptation to climate change in forestries. So I'm a population geneticist, so what I'm pointing today is mostly population genetics methods and also a bit of quantitative genetics. So, and without more introduction, I will start. So I will start by explaining the context in Europe, where we are working so much right now into what is the interest of working with the population's responses of forestries to climate change, and we are getting ready for the future that is coming. That's where we are expecting very huge changes in the distribution of species and in the responses of forests to the environment, and also we have some policies in Europe right now that we are planning for a huge effort in reforestation. So you may have heard about the European Green Deal that has one of the mechanisms that are foreseen is the planting of at least 3 billion of trees by 2030. And one standing issue right now is how to do that, how to do this plantation, how to source the forest reproductive material that we will be using. So on some answers about this, it comes from modeling approaches to assess the population responses to future climates. And there are some traditional methods. Actually, we are lucky in a way in forestries because we have a long history of common gardens on coordinate trails in experimental plantations where we are comparing populations coming from different origins. And this has been used for quite some time now to model the response functions of these of this population, so how they will behave when they are transferred from one environment to another. And this is very useful information for knowing how well, not only how natural forests will respond to future climate, but also when we are developing reforestation or plantation scheme to know what would be the best point answers to plan thinking not only in present climates, but in future climates. And now we are in a very exciting moment in forestry because we are beginning to have proper genomic resources with many reference genomes for several forestries, and this could give us very good opportunities to integrate genomics into this standard, more traditional tradition framework. So, what are the advantages of including genomic data in this kind of predictive models? On one side, they are more informative. They allow us to understand better what are the drivers of the genetic response at different levels. Then it allows us also to consider the interspecific variability, the divergence that we found at the different populations across the distribution range of a species in much more detail than just the traditional modeling approaches. It gives us also higher capacity to do predictions for a larger number of species and populations, including outside the populations has been genotyped or characterized, or they have been tested in common gardens. Most importantly, this is fast. So it's much faster to do a prediction based on genomics than to plant a common garden and wait for 20, 30 years till trees are adult, they grow, and we can have good phenotypic estimations. So, and there are different methods to integrate genomics into these prediction ecological models, but they from very simple to more complicated ones. So I'm going to do just a brief introduction, and this is a field that is actually changing very fast and moving forward very fast as well. So okay, so a simple way for considering genetic information is just to consider that distinct gene pools. That means that different population may have different also adaptation responses to climate change of any other environmental change, and for that there have been already some work showing like this paper in bats that's not trees, but in trees work similarly, like they can get very different predictions of future distributions if they consider models not by considering the full species at the same time, but they do different models for different ecotypes. And that for forestries in Europe, and I guess that for most plants and many animals as well, this can be already useful because we have it's typical to have quite substantial population genetic structure. So we have very often populations that have had different performance and that they have a different history, and they have been isolated for thousands of generations, and they have evolved differently. So it makes sense to do models separate for them, and that changed a lot the predictions. Actually, I did not bring any studying for the street to the talk, but some colleagues working in these approaches like are finding that the prediction, when you consider the natural genetic variability that there is in the most forestries that are generally undomesticated species, you find that the predictions are not so gloomy actually those that you get when you consider a species as a whole. Okay, but that's actually what is now becoming very popular is rather not only considering this kind of I would say soft genomics, we just use genetics or genomics to just identify gene pools, and then you do modeling for the different gene pools, like we use traditional methods, but then we are starting to produce models or ways to estimate or components of population vulnerability that are just based on genomics. And you may have heard or read about the genomic offset methods. This is becoming actually extremely popular, starting in 2018. There have been already several papers that that are using these methods to give a sense of how natural forests could be affected by climate change, and to identify regions that could be that are less adapted, or they could be less adapted to future climates. And how this works is actually the idea is well, it's not very, it's quite simple, I would say, but it takes some work to. So I will navigate you through it. So first we start having genomic data and environmental data that is typically is climatic data, but any kind of environmental data works. And then we have first to establish correlations between gene allele frequencies and different parameters, climatic parameters, for example. So like alleles that are correlated with temperature. And then with that, what we can do is to, on one side, we have the current climate. So on the current genomic composition of the particular population, and on the other side, we can do a prediction of the future climate, and we can also identify using this gene allele frequency correlations with climate. We can also identify or know what could be the optimal allele composition that a population considering the future climate. So the genomic offset is just the difference between this genomic composition that we have right now in a population and that could be optimal in the future. And that's something that is, as I said, getting very popular. And as we will see when I'll point some of the case studies we are developing, what is really important is to have some independent data where we can do some validation steps. This is just predictions based on genomics, and they have quite some strong assumptions, so it's good to have some data that it can be either data from the field, like this paper. This paper, what they have is inventories of presence, abundance of the species, and then they correlate the genomic offset with the abundance of the species to say, and they really find that in the populations where this genomic offset is higher, they have less abundance, and that suggests that this is because climate change is already taking place. So these populations are maladapted, and this is reflected in the demography of the species. And in the case of trees or other plants or crops where you have common gardens and on comparative experiments planted in the field, you can use these common gardens to correlate the genetic or genomic offset and with some traits, and find out, like in this case where they have the highest genetic offset, the difference between the current genomic allelic makeup of the population and compare with the future one, then the lower the height for those trees in a particular common garden. So I would say actually that common gardens are very useful and very helpful. And most of the approaches actually that we are using for integrating genomics into ecological models or prediction models of population performance in natural conditions. They are based on previous genome-wide association, GWAS, that use adequate panel where we have genotype and phenotypes, and we can compute things like polygenic scores or positive effect alleles. That is a way of collecting the information we have from phenotypes into genomic data. So the polygenic scores that probably some of you or most of you are familiar with polygenic scores. It is just the allelic effects of a particular allele, a particular marker on a phenotype that is weighted by the allele frequency. And this gives you, when you do the summatory of many markers, then you have a score that actually can replace the trait itself. And a more simple way of summarizing this information would be to use what is called positive effect alleles. That is, this is just counts, simple counts of polymorphism that have non-zero effect on phenotype. So assuming that if a particular individual has many of these positive alleles, is going to grow more than individuals that doesn't have these alleles. So, and one thing we have learned in forestries and I would say in many other organisms, starting with the first studies in humans, is that most adaptive traits are polygenic. So most of the approaches that are based on candidate genes are actually doomed to fail because most of the traits that are important for adaptation, actually they are controlled by many polymorphisms with very small effects. And that's what makes like there are still some genes and some alleles that have major effects, but we find out also that for most traits, most of the variation is polygenic. So actually what we need is to find methods where we can integrate many markers and that have very small effects into a prediction model. And that's one of the methods we are using for doing these predictions and also for computing the genomic set. They are moving from methods based on candidate genes to methods that are polygenic, that just use multivariate approaches. So, and to finish my introduction, just to well, not only an introduction, but the overview of the approach. The other thing that is very important for us is to identify the relevant traits that are in the selection. That means traits that different populations have different adaptations. So and these adaptations, they are associated with particular climates, and there are traditional methods like the comparison of QST with FST. So FST is just the genetic differentiation as you compute it with molecular markers and that are neutral, and they are supposed to just show you demographic processes and divergence due to isolation of populations, while QST is computed based on additive variance and quantitative traits. So if you find a difference that QST is higher than FST, that is expected to mean that this trait has more adaptive divergence that's expected by the neutral processes such as demography or isolation. But these methods that are the standard methods, they are not very useful for species with very complex population genetic structure. And now we are trying to implement, building on some work that was done a few years ago, but the group for Graham Coop in UC Davis, we are trying to develop these other methods where what you have is basically principal components analysis, a principal component analysis that can account for properly for population structure and differences in differentiation across populations. And what you do is to project the traits, the quantitative traits on the species. And you can see here in this figure quite clearly. First, you have in this the axis you have the you have projected the populations, and you see the different colors mean different gene pools. And then in this axis here you have a quantitative trait. In this case, is the specific area. And then you can see that using these approaches, you not only identify when there is a significant is a higher differentiation for the quantitative trait than expected just by population structure, or by demography, or by other process not related to adaptation, but also you can see which populations are responsible for this divergence. And you see this line here that is showing these values. You can see how it's basically this what is a little bit confunded with the legend, but is actually this color here, the gene pool of trees from Morocco, that actually they have very high specifically area compared with these other trees that come from central Spain. That's what is telling you basically the adaptive divergence for this trait comes from having these two different gene pools. So, and for the rest of the talk, I will be talking about Maitland pine, that is our model species in Bordeaux. Is one of the model species in Bordeaux, and this is a species that have a huge economic impact in the region, where we have over 1,200,000 hectares of plantations of Maitland pine, and also it's a very important in the natural forests in all the Mediterranean area or southwestern Europe. So, and for these species, we have a network of clonal common gardens that are quite huge. So we have a very large phenotypic data set, and pretty modest. We have only so far 10,000 SNP markers for the species. That is a modest genomic data set, but this is enough for the so far it has proved to be enough for the kind of work we are doing. So, I will introduce you to Adelaide Teradot, that is my PhD student in INRAE, and his PhD student shared with another researcher from the Slovenian Forestry Institute in Slovenia called Mariana Westergren, and she's working with this data, with the Maitland pine dataset, and she has just produced a very complete map of the population structure in the species. You can see that we have many gene pools coming from Morocco to the region where I live in Bordeaux, and also Portugal, Italy, some islands in the Mediterranean. And this genetic structure makes modeling complicated, but also brings some advantages, like we will see, because it normally represents different phenotypes as well. And that's what we see. We see that there's a really big difference in phenotypes, and also in genotypes, in growth traits and other traits for the different gene pools. And this is a picture I took a few days ago from one of the common gardens in Bordeaux. You can see these two trees have exactly the same age. They are planted in the same place. The only difference is that this tree comes from Morocco. This other tree is a local tree that comes from Bordeaux. So a local tree. So, and with another young colleague in our lab, Juliet and Chambo, that is a postdoc with us, we have produced a first study where we are, Juliet is a modeler, so, and she has a very strong expertise in ecological modeling. So the idea here was to use like the standard model for prediction of population responses and try to improve them by adding genomic information. So what she did, and this has just been published in American Naturalist just in October last year. So she built 12 different models from the standard ones to others that includes both the climate of origin of the populations for which we want to do the predictions, and also some genomic data we have from these populations, and then also models that use only genomics. And this is done in a common garden. Actually, we are doing predictions. We are developing a method to do these predictions, but we can validate these predictions because we have the real data. We can measure directly the phenotypes in these common gardens. So what is new from the traditional modeling? So what is new is that we are including from the information we get from the genomics we have done, or the genotyping we have done on these trees. On one side, we can include the gene pools, the information we have about each population, to which gene pool the population belong. And also we have computed, we have doing a GWAS, an association study, and we have identified alleles. Actually, we have a polygenic association study. We identified many alleles that have non-zero effects on the phenotype. And then we have just done counts of these alleles, and we introduce these counts, this number of alleles that have a positive effect into the models. And we have two kinds of counts here. On one side, as for this particular species, we have 5 common gardens. So when we do the GWAS, we compute what is this global PIA, that is this global positive effect alleles. That is when we consider all common gardens together. That means that we are trying to identify SNPs. We are trying to identify alleles that are associated with the phenotype for when we're considering experimental plantation all around the distribution of the species. But then we are also testing what we call regional positive effect alleles. That is when we just choose AGWAS where what we identify is association in a particular common garden. And so this is associations that are unique and specific for a particular environment. So they may not work outside this environment, but they have quite some explanatory value in locally. So Juliet ran all these models, and then what we found is that our best model were including on one side the gene pool. So actually we have already a lot of predictive power just by knowing the gene pool where this tree came from. And then actually we got very good predictions when we integrate this genomic information together with the climate data. So actually what is most interesting is that we get even better predictions just with the climatic data of the populations, the climatic data of the origin of the populations, and the genomic data that actually when we analyze the common garden in the traditional quantitative genetics model. And also what we found very interesting is that the better prediction comes not from these global positive effect alleles, but for the regional ones. What means that actually what we are finding, what we have, at least for our case study, is more common to have polymorphisms that have a regional value, that are predictive at a regional scale, but that they are neutral outside of that region, and that contributes to a quite long-ranging discussion about how polymorphisms affect phenotypes under two contrasting models on one side, there is the idea of that polymorphism may have antagonistic pleiotropy. That means that SNPs or alleles that have a positive effect in one part of the range and a particular environmental conditions will have actually the negative effect in other part of the range. So, but that means that they are important in other range. And there is an alternative explanation that that says that is called conditional neutrality. That's where we can have a polymorphism that explains is positive in one particular part of the range under some particular conditions, but this same polymorphism will be neutral outside this range, or even at another time. So that says that the adaptation value of polymorphisms is something that is not absolute, but it depends very much on the conditions. So, and the work we did is supporting that is more these regional associations that are important. And finally, we have also started to work with the genomic offset approach in maritime pine, and it's actually an approach that is very appealing because if you look to identify forests that have maladaptation risks, and it only uses genomic and environmental data. So that means that you save a lot of time. You don't need any phenotypic information. You don't need any common garden, and from the practical point of view, is actually this kind of methods that can help a lot to identify what are the most adequate provenance for plantations or for natural enrichment of natural forests to make them more resilient in the face of climate change. So, and if you know this method, you have followed the literature. They are really getting very popular for kind of species. So that means there are several methods currently implemented, and that there are a lot of case studies coming from different species. But what we are finding in our own experiences that is not so obvious, because first you need to identify these robust gene environment correlation, these associations between allele frequency and a particular variable, climatic variable, or the environmental variable. This is what we find, and this is our own finding, but there are many people finding the same. Actually, when you try to do these associations with the environment, every method identifies a set of markers, a set of polymorphism, but there are very little overlapping between methods. So it gives you quite, on one side is kind of expected, because if adaptation is truly polygenic, then there will be polymorphism with very small effect, so there will be very difficult to identify with population genetic methods unless you have a huge sample size, and also each method has their strengths, so maybe they are identifying some but not others. But this is a very big complication for the genomic or self approaches. And then even if you come with a way to identify these kind of genes, then we are using in our study 4 different methods to compute genomic offset. And what we find is that there are some methods like the ADA, LFMM. They are correlated, so this is very nice. We have 2 methods that are giving you the same positions. But then there are others that actually you have negative correlation. So using a method gives you a prediction, and using another method gives you the inverse, the contrary prediction. So this is actually gives you very little confidence on how valuable these predictions are. But we're still working. This is ongoing work with Juliet, and we're still working in finding the best methods. And actually, that's one of my main conclusions that we will see. we cannot, it was our dream to use only genomics, not to use the common gardens, but we still need the common garden for validation of which methods are working better in our particular case study. So then I will just finish with some conclusion and perspectives. So what we have found is that integrating genomic into population models of population response to climate actually is good. It improves the accuracy of predictions, and that's helpful because it's giving valuable information. We find that when we have these models that combine multiple factors, but that they are actually based on the traditional model, but the genomics can improve the accuracy, but it's not the only component of the model. And in these models, the genomic is useful, but actually we would need to develop a different model for each environment, because there's a predominance of polymorphisms that are conditionally neutral. That means the polymorphisms that they are important just in particular environments. Then when we have trial of genomic-only approaches, such as those related to genomic sets, we found that they are useful, but it's not very obvious to identify which method is working and which method is not working. So still, you need some kind of field data or some common garden data to do a validation that is independent, that is different from the genomic data. So that's okay. For the conclusion that for us, at least for the trees, we still need these common gardens, and actually we need it more and more because we will need to. Once we develop one of the prediction methods, then you can use it just with the genomic data. But at least at the beginning, you will need to have common gardens for all kind of species and provenances and populations, and test it in a very wide range of environments. And this is my previous to last slide, just to say that most of the projects that are currently using genomics are kind of static. That means that if we consider vulnerability, and this class comes from one of the IPCC reports, then actual vulnerability is composed from exposure to the threat, that is the future climate, and the sensitivity, how the populations actually, if they are more or less sensitive to these changes, but there's also a capacity for adaptation. And methods like the genomic set, they consider that they are a correlation, a relationship between allele frequencies and climate, or climatic parameters that doesn't change, that is static in time. But we know that there is adaptation, and not only adaptation, there is also plasticity, there can be migration. So what we need, and I think what we are working in our lab towards developing some additional framework where we can integrate not only the genomics, but also the capacity of natural populations to adapt dynamically in real time to the changes that they are coming. And with this, I would like to just to thank you so much for your attention, for listening to my talk, and also to say that we are now this work. This is a work in progress, and we were lucky enough to be supported by a new European project that just started, that is called Oak Forest, and that will provide a lot of support and funding for this kind of modeling that integrates genomics from here till 2027. So thank you so much, and I will be very happy to take any questions.
Thank you, Santi. Thanks for the good talk.
Thank you.